Welcome to Library Seminars, NOAA Central Library's webinar series for the presentation of research and ideas that reflect the National Oceanic and Atmospheric Administration's mission. The seminar is part of the Knauss Lunch and Learn series, which is a monthly webinar where the 2022 Knauss Fellows showcase their own research, like today, or the projects they're supporting during their fellowship. Today's presentation, Coral Symbiosis Under Future Ocean Conditions, will be presented by Hawaii Sea Grant Knauss Fellow, Mariana, Rocha, de Souza. Caitlin Lang, a 2022 Knauss Fellow working in OAR's Office of the Assistant Administrator, will be our host and will introduce our speaker. But before I turn this webinar over to the fellows, here are a few tips to help you enjoy this presentation. The webinar is being recorded and will be available on the NOAA Central Library YouTube channel later today. I will put the link to the channel in the chat box once the presentation starts. We highly encourage you to ask questions, which the speaker will address at the end of her presentation. So please type your questions throughout the seminar as they come to you. The question chat box where you would put these questions is located in the GoToWebinar control panel, most likely on the right side of your screen, represented by an arrow. And with that last detail, I'm going to turn the presentation over to our host, Caitlin. The mic is all yours.
All right, thank you, Lisa, so much for that introduction. I'm thrilled to have the opportunity to introduce Mariana Rocha de Souza, who will be giving a talk today at the Knauss Fellows Lunch and Learn series. I want to thank Lisa Clark and Knauss Fellows Giselle Schmitz and Violet Doucette for helping organize today's talk. So Mariana is from Brazil and got her PhD in marine biology at the University of Hawaii, Manoa, where she studied the interaction of corals and their algal symbiont and how they respond to climate change. She is currently placed at the Global Ocean Monitoring and Observing Program at NOAA. She loves hiking, diving, and recently adopted a schnauzer named Neppy. So without further ado, Mariana, please take it away.
Thank you so much for the introduction, Kentley. So hi, everybody. Thank you all for coming. I'm Mariana Rocha de Souza. I'm a 2022 Knauss Fellow. And as she said, I'm placed at the Global Ocean Monitoring and Observing Program at NOAA. And I'm very glad to be presenting you the work that I did for the last couple of years as part of my PhD in the Coral Resilience Lab and in the Tobo Lab at the University of Hawaii, where I studied the effects of climate change in the corals and in their symbionts. So corals are among the most biologically diverse and productive ecosystems on Earth. They host about 1/3 of marine species. They provide incredible cultural and ecological value. They protect the coast against the action of waves, and they inject billions of dollars into the economy as source of fisheries and tourism. And corals are colonial animals. They are formed by many polyps. And in each one of the polyps, you can see millions of cells of a microscopic algae from the family Symbiont. This symbiotic interaction of the corals and the microscopic algae is fundamental for the survival of corals. The corals and the algae have a mutualistic relationship, similar to other relationships found in nature, where both organisms benefit from it. So the corals provide protection to the algae, and the algae photosynthesizes and shares the product of photosynthesis with the corals. And it's responsible for about 90% of the coral energy requirements. Unfortunately, the burning of fossil fuels has led to the increase in CO2 in the atmosphere, causing climate change, and increasing the global overall temperature. And the ocean absorbs heat. A recent paper from 2020 estimated that only in 2020, the ocean absorbed 14 gigajoules of energy. So that corresponds to about 7 Hiroshima bombs being exploded every second. And in fact, the sea temperature in most of the tropical regions have been increasing by about 1 degree Celsius over the past 100 years, and are currently increasing at 1 to 2 degrees Celsius per century. And this increasing temperature in the waters results in the breakdown of interaction of the coral and the symbiotic algae, the algae that I just mentioned. So the symbionts, when the temperature of the water gets too hot, this interaction breaks down because the symbiont starts producing too much reactive oxygen, which is toxic for the corals. So the corals lose their symbiont and then lose also the main source of energy. And this phenomenon is known as coral bleaching. And coral bleaching has been increasing. So we've seen an increase, a 5-fold increase in coral bleaching events since the 80s. And from 2008 to 2019, we have lost already 14% of the global corals. But in a bleaching event, no corals respond in a different way. So corals are being affected by climate change, which is causing bleaching events worldwide. But in a heat stress event leading to a bleaching event, not all corals are going to bleach in the same way. Like in Hawaii, when I used to snorkel, you can see during a bleaching event, corals side by side, and one is bleaching, one is not bleaching. So individual corals can respond differently to this stress. And this is because there are few mechanisms for coral resilience. There are few mechanisms that can make coral more respond differently to stress. So one of those mechanisms is the community of algal symbionts that the corals have. There are different species of algae, and the type of algae, the algal symbionts that the corals have, can influence how corals are going to respond to stress. For example, the symbiont from the genus Cladocopium, that's the generalist symbiont in Hawaii, that's the most commonly found symbiont in the corals in Hawaii, it's a generalist, so it's just found everywhere. The symbiont from the genus Durusgenion, the clade D, is considered to be a heat-tolerant symbiont. So if corals have this symbiont, they usually bleach less, they become more heat stress resilient. And symbionts from the genus Symbiogenion, clade A, or Breviolen, clade B, they're considered to be opportunistic. They are parasites. So they're found in corals when their immune system is compromised. And they're also found in corals, has also been associated with coral diseases. So the community of symbionts can affect how corals respond to stress. Actually, bleaching can be a mechanism for corals to change the algal symbionts that they have to symbionts that are going to be more resilient to the stress events. And this change can happen in two ways. So it can happen by shuffling. So it's just like a change in proportion of the symbionts that already existed inside the corals. Or it can also happen by shifting. So the symbionts are acquiring completely new set of symbionts from the environment. So the resolution of the techniques used to identify the symbionts can make it hard sometimes to see what's actually happening, if it's shuffling or shifting. But no matter the mechanisms, identifying the algal symbiont in corals and how they respond to heat and other environmental stressors is very important if you want to be able to predict how corals are going to respond to the future ocean conditions. So the community of symbionts hosted by corals has an important role in the coral physiology and in the response of corals to the environmental stressors. So for my dissertation, I decided to quantify the algal symbiont community in corals and investigate the main drivers, what was driving this community composition in corals by combining field and long-term mesocosm experiments. So combining all these results, we can get a better understanding on the role of symbiont community composition in corals and providing resilience for corals in the climate change for the stress they expect to see in the coming years. So we know corals are being affected by heat stress, but heat stress is not the only stressors affecting corals. The increasing CO2 in the atmosphere leads to the increasing heat, as I said, but also a decreasing pH, an acidified condition. So that led us to the question of how Hawaiian corals are going to respond to those conditions in the future. We then designed a mesocosm experiment with tanks. So we had 40 tanks. When we exposed corals to acidified and a heated condition, and the acidified and heated condition is expected by the end of the century. And we sampled the corals to look at the symbiont community before the heat and the acidified condition treatments and after being exposed to these treatments. So just like more detail on the experiment. So we exposed fragments of 8 species of corals. So we had 3 of the main genus of corals in Hawaii. So for Montipora, we had Montipora captata, Montipora flabellata, and Montipora patula. For Porites, we had Poraitis compressa, Poraitis evermanni, and Poraitis lobata. And for Poecilopora, we had Poecilopora acuta and Poecilopora evermanni. And we exposed all these coral species of the main coral species in Hawaii. We exposed them to 4 treatments. So we had the control treatment, so no stress, acidified treatment means lower pH, heated stress, so higher temperature, and then the combined stressors of acidified and heated conditions. And we exposed all these fragments of corals in these treatments for 2.5 years. So it's a very long experiment. And the corals we collected came from different locations. We had 6 locations in Oahu, and we chose these locations because we wanted to have corals coming from very different conditions of temperature, nutrient input, pollution, and because I know not all of you are familiar with these regions in Hawaii, I'm gonna just break down a little bit, give more details about these locations. So we had corals coming from Kaneohe Bay. We had 2 locations in Kaneohe. We had Sampan Channel and HIMB. So HIMB, that's the Hawaii Institute of Marine Biology, that's in the south of Kaneohe Bay. It's a region with lower water circulation, a lot of water runoff from the mountains, and it's a very coastal area, HIMB. Sampan Channel, on the other hand, it's a more open water, open ocean location. And we also had corals from Waimanalo. Waimanalo is a beach in the south, like sandy beach, high sedimentation. We had corals from Magic Island. Magic Island is a highly urbanized area. It's close to Waikiki, for those that have been in Oahu, and frequently it has rain-driven effluent and increased nutrients from the Alawai Canal. We had corals from Electric Beach. It's a very famous tourist beach for snorkeling, and the corals there are exposed to high temperature due to release of water from electric power plant. That's why the name Electric Beach. And we had corals also from Haleiwa. So Haleiwa is in the North Shore. That's one of the sites for where one of the main competitions, surf competitions, happen. So it's a very turbulent site and known for like a lot of like water movement, big waves. So also giving some background here about like how I identify the algal symbionts. So the algal symbionts live inside the cell of the coral. So once we select a colony we want to study, we cut a small fragment of that colony. And inside the fragment, there will be DNA of the coral, of bacteria, of the algal symbiont. So like anything that's inside or at the surface of the coral will come with that fragment. We then extract the DNA of that fragment. And as I said, we're going to find the DNA of everything that's there. But then we target, we make what we call DNA libraries. So it means we're going to separate, we're going to target only what symbiont DNA. And we're going to make copies of those DNA and prepare it for sequencing. And then after sequencing, we get files with the nitrogenous bases, like we see here, like the GACT. And the next step is to make sense of all of these bases and identify the different symbionts present in the coral. So we use SymPortal. SymPortal is a portal specifically designed to analyze symbiont ITS2 data. And then we get information of the number of DNA copies and the proportion of the different types of symbionts in each coral. Still giving some background, so just because I don't know how many people are familiar with the visualizations that I'm going to present. So there are multiple ways to visualize the symbiont inside the corals. And one of the ways we can visualize the different types of symbionts in a coral is by doing a distance-based analysis, where each point represents the symbiont community in one coral. And if the symbiont community is similar, the points are going to be close together. And if the symbiont community is very different, they're going to be far apart in this space. So let's imagine here we have 2 corals, and then one coral has the symbiont A and B. The other coral has the symbiont A1 and B. So the symbiont communities are very similar. So therefore, these points are very close to each other in the space. Let's say we have another coral right now that has the symbiont ABC. So the symbionts here are a little bit more different. So they're going to be a little bit more far apart, more far from each other in this space. Let's imagine we have other corals of completely different types of symbionts, like C and D. So they're going to be even farther. And then we can add ellipses to these groups representing 95% confidence intervals of the coral members of each group being inside the ellipses. So jumping into the results, just get some water here. So I'm going to be presenting the results in two groups because we have a lot of variables. So we had site, we have treatments, and we have coral species. And we also have the different time where the different times when it was collected, right? Like the initial before being exposed to treatments and after. So I'm going to be presenting this, in different groups. So I'm going to be presenting the initial collection. It means prior to any treatment. I'm going to be presenting the results for the control. It means no stress at all. So like just ambient water temperature coming from Knauss Bay. And stress treatments of acidified, heated, and acidified treatment. So I grouped these stress treatments with all this stress together to be able to visualize. Otherwise, it's just too many things to look at a single time. So just keeping that in mind when I'm presenting. So again, here I'm presenting the initial collection. It means before being exposed to any treatment, control, no stress at all, and then treatments. It means all the other remaining treatments combined. And here I'm showing the results of the Symbiont community in each one of the species of corals. So remember, I had 3 genus of corals, but 8 species in total. And we can see that previous to the treatment exposure, so at the initial collection, the Symbiont community was more similar among species from the same genus, and that after the treatment, the Symbiont composition was very species specific, with corals from each species hosting a unique Symbiont community that is significantly different from corals from other species. It's also very interesting to see that the confidence intervals, it means the size of the ellipses, for each species is very different. Porites, for example, here represented in red, more like warm colors, is known in the literature to host one single type of symbiont. So you can see here in the ellipses that the confidence interval is very small. So like very small difference in the Symbiont community in that genus, which corresponds to what we have been found in the literature. But it really contrasts, for example, with Montipora. And we know Montipora captata has been known to host C and D, so like multiple types of C, multiple types of D. So can host a very diverse community of symbionts. And you can see that here in the ellipses. So it's very interesting to see how the different species can host different types of symbionts. Here we're looking at the different sites, so how the treatments have affected the different sites. So here, all these species are grouped together. We're not looking at the different species independently, but all the species are grouped together, but we're looking at the difference per treatment. And we found that the site of origin influenced the Symbiont composition, and that the initial collection, previous to the treatment at the initial collection, only corals from Magic Island here are different from 2 other sites. But after being exposed to the control treatment, it means no stress, the different locations have a more similar Symbiont composition. So there was no difference here after the control. So after being exposed for a long term in the same conditions and no stress, most of the locations had a very similar Symbiont composition. But what is very interesting, I think, is that after being exposed to the treatments, so 2.5 years exposed to stress, we still see a signature of treatment. So even after staying for that long, for 2 years, in a heat stress condition, we still see a signature of where the coral came from. So we see here, after being exposed to the conditions, that Knauss Bay, there you go, Magic Island was significantly different from Knauss Bay and HIMB. And this really highlights the role of environmental memory in the corals. So like the long environmental conditions corals are exposed to in their site of origin creates in corals what we call an environmental memory. It's like a memory of stress conditions that makes corals from different locations respond differently to stress, even after 2.5 years in the same mesocosm conditions. There you go. And while corals were exposed to different treatments of temperature and acidification, here we're looking at the treatments. What's the effect of treatment in the symbiont composition? We found that low temperature treatments, which are the control, the initial collection, and the acidified, had a similar symbiont composition, and corals are exposed to heat stress, which are the acidified, heated, and the heated. They had a similar symbiont composition. So really means that the heat stress is what is driving the symbiont composition here. Acidification was not a main driver. So even though we know heat stress and acidification are going to be the conditions expected in the future, it seems that acidification is not what is driving the symbiont, that heat is going to be the main issue for corals. And then finally, I'm going to be showing the results of the symbiont composition itself. So I grouped them by kind of their main group. So I'm not showing the subtypes, but I'm showing the main groups here. And I'm showing only for 3 species, 1 from each genus. And I highlighted in orange, so like a group in orange, the heat stress conditions. So like the acidified and the heated are highlighted in orange. So looking at the symbiont composition, I'm highlighting here only 1 example from each one of the genus. As I said, Montipora capitata, Porides compressa, and Postulopora acuta. And we can see that for most of the genus, we see an increase in heat-tolerant symbionts. I'm trying to find my arrow. There you go. We see an increase in heat-tolerant symbionts in the heat stress conditions. So the D1 and D4, like in warm colors, you can see an increase. So when corals are exposed to the heat stress, as expected, we see an increase in heat-tolerant symbionts in those conditions. It's surprising to see that in some of the species. So like Porides compressa and Postulopora acuta usually don't host this type of symbiont. And we think that might be because these corals have been exposed for so long in these treatments. So maybe that gave them time to actually acquire these symbionts. But it's also interesting that in the same conditions in the heat stress, we also see an increase in opportunistic or parasitic symbionts. So we see an increase in B1 and also A that are considered opportunistic and parasitic. And we think that, yeah, like it also makes sense. Like it seems that in those conditions, the immune system of these corals became compromised. And then corals just acquired, they're not able to select what type of symbionts they're acquiring anymore. So they just become more vulnerable to the infection of different symbionts. There you go. I should have clicked before. And when we plotted the diversity of symbionts according to the number of reads that we are getting for each sampling effort, we see that even increasing the sampling efforts, at a certain point, the symbiont diversity gets saturated, reaches a plateau. And we see that overall in the warm treatments, I'm gonna click now, in the warm treatments, acidified, heated, and the heated, there is an increase in there is a decrease in the symbiont diversity. This could suggest that corals with higher symbiont diversity, or they died, so previously to our collection, or it could also suggest that corals selected for heat stress tolerant symbionts. So just a recap, also gonna get our water. Just a recap of everything. So we're looking here at the symbiont community in corals in Hawaii. We had 8 of the most common species in Hawaii, and we exposed these corals to the conditions of acidified and heated in combination, but also individual stress conditions. And we also had a control. And we sampled these corals before the stress events, before being exposed to these treatments in the tanks, and after 2.5 years being in these tanks in those conditions. And we found that the Symbiont species is very specific by the coral. The Symbiont species is very specific according to the coral species. So we saw that after being exposed to these treatments, in the end of the experiment, each one of the coral species had a very different Symbiont community composition. We saw that heat is a main driver, but acidification doesn't seem to be a main driver of the Symbiont composition. And we know that with the literature, we know that bleaching is mainly happening because of the increasing heat temperature, increasing temperature, like the heat stress. So it seems that really acidification is not going to be the main driver. It's going to be affecting corals that much. We also saw that site of origin matters. So even after being exposed to the conditions for 2.5 years, we saw that the location where we collected the corals it still had a say in what Symbiont communities were present in the corals. So like it really says there is a really shows that there is an environmental memory in the corals. So like the Symbiont community in the corals is dictated by where the coral came from. Like the stressful conditions that these corals were used to previous to being exposed to the treatment conditions in the tanks. And we also saw that there is a decrease in the Symbiont diversity during heat stress. So like after we had these 4 treatments, we see that the Symbiont diversity really decreased in any of the heat stress conditions. So it could be that corals are selecting for this symbionts, or that we just lost all the corals that had a more diverse symbiont. So to conclude, I know it's a lot of different results and different factors here, but I think together these results really highlight the complexity in predicting coral response to future ocean conditions. And we have to take all of this in consideration, right? Like different species, different locations, and the different conditions in each one of the sites where we're collecting these corals. And the algal Symbiont community composition of corals in my work changed in response to their environment. And this response was dependent on both the coral species and their site of origin, highlighting the role of symbiont specificity and the environmental memory in shaping coral resilience. So with that, I want to thank all my collaborators, so all my lab mates and interns that helped this work come true, from the Coral Resilience Lab and the Tobo Lab in Hawaii. And I also want to thank all the funding agencies that allowed for the sequencing, for all the field trips. It's a lot of work, and yeah, very glad to be done with all of that. And with that, I'm open to take questions. Also open to for people, if people prefer to have questions by email, just feel free to contact me.
Awesome, thank you so much, Mariana. So audience, this is your time to ask questions. We'll take the next few minutes to do that. And if you do have a question, please type it into the chat box, which is part of the GoToWebinar control panel, most likely on the right side of your screen. And while we wait for your questions to come in, please remember that the recording of today's webinar will be uploaded to the NOAA Central Library YouTube channel in the next few hours. So if you would like to share Mariana's work with a colleague, we encourage you to visit YouTube and share the recording's URL. And I'm about to push the send button, so you should see that link in the chat box. And also, before we start taking questions, feel free to download Mariana's slides from the handouts menu in the control panel. They were beautiful and informative, and they have her contact information. So this is the time to do that before we go offline. And just, I see comments coming in, so lots of compliments, Mariana. Very interesting. And someone asked, wow, is that you in this picture?
Yes, it's me.
There you go. All right, so we do have a question here. It says, thank you so much for your talk, really great work. I was wondering if it would be possible to use your results to rank the sites of origin in terms of their tolerance to heat stress and ocean acidification.
Yes, sure. That can be something that can be connected to the resilience, right? Like there are multiple things, I think. So resilience is not just the symbionts, right? Like, so corals have the interaction of the symbionts, and the symbionts they have an important role in the resilience of corals. But the resilience is not just driven by the coral community. That is also a genetic component, right? Like, so, yeah, as I said, when we're swimming, many times I would be swimming in Hawaii, like during a stress event, during a bleaching event, and you can see side by side corals that one is bleached and one is not bleached, and they're from the same species. And we can look, sometimes the Symbiont are different, sometimes it's not. So there is also other factors controlling the resilience of corals, not just the Symbiont. It can be bacteria, it can be the genetic of the coral itself. Different species also respond differently. So the Symbiont has an important, is an important component for sure, but there are multiple components in providing coral resilience. But yes, for sure, like that can be something that can be used to understand, yeah, which sites, what conditions are present in each site, and how we think corals are going to respond, but not just quantifying. Just quantifying this, the algal symbiont is not going to give a full say of how corals are going to respond, because the genetic of the corals also matters a lot. But thank you for the question.
Yes, actually, Caitlin had a question. I don't know if Caitlin, if you want to ask it personally, I'll give you a second to pop on. If not, I will happily read this.
Yes, so let me just pull it up really quick. So Mariana, thank you for that talk. That was awesome, and I actually learned something. And it's, I always think it's nice to hear what the fellows studied, prior to coming to DC and what their background was in. So as people look to not only conserve but restore coral reefs, what recommendations would you give them? So, for example, should we prioritize restoring coral reefs that are heat tolerant, or restoring all biodiversity, even if they might not take as well as the heat tolerant corals, or should we just prioritize restoring corals in cooler areas? So I don't know, but I'd love to hear your answer.
Good question, very good question. So yeah, there are a lot of initiatives on coral restoration, like coral gardening was very popular, it's still very popular. And I think before, like some years ago, the idea was just like, let's collect all the corals, like fragment them and then plant them. So like after any stress event, they would just go and plant corals on the reef. And these corals would survive until another bleaching event would come and then kill everything again. And then like you have to come back and do the same effort again. So something that we have been doing in Hawaii, like in the Coral Resilience Lab, is coral restoration, but with corals that we know that are already more resilient. So as I said, the genetic of the coral dictates also like how corals are going to respond. So we would go to the reef, collect a few corals, bring them to the lab and expose them to heat stress. So testing how far can these corals go, and then which corals are gonna survive. So like which ones are the corals that are already more resilient, which corals are already stronger. So selecting these corals that we know that are already stronger, and then outplanting them on the reef. So like they have a higher chance, they're gonna have a higher chance of surviving any bleaching event. One of the issues is genetic diversity, right? Like we can, that we have to take that into account. Like by doing something like this in the lab, we have to try to get as many corals from, as many genetic diversity, as much genetic diversity as we can. So like really selecting different individual colonies and then testing these corals in the lab and then outplanting them. And yeah, trying to keep the genetic diversity, because otherwise that can also affect how corals are gonna survive any other stress event. They might be resilient to bleaching, but maybe they're not gonna be resilient to any other stress that will come if they have such low genetic diversity. But I think that's a very good strategy. Like I think bleaching is the main thing, like heat stress is the main thing affecting corals, and it's gonna be persistent until we actually fix climate change. So heat stress events are going to keep happening. They have been increasing in frequency. Just my time in Hawaii, we had bleaching in 2014, 2015, and 2019. So like the frequency is becoming like it's becoming more and more frequent to have bleaching events. So I think that's the main thing we should address. So I think trying to find corals that are more resilient and then doing restoration of these corals that are more resilient, that's the best strategy.
Thank you so much.
Yes, thank you. And we actually had a similar question, but I'm going to ask it in case you have anything to add. As a compliment first, it says, great talk, Mariana. I'm wondering how you might apply this work on coral symbiont responses to temperature/acidification stressors in coral restoration projects.
Yeah, great question. So as I said, like the resilience is multiple things are going to dictate the resilience of the corals, and symbionts also have a say in that. So like understanding which symbionts are present in each coral and what's the role of the symbiont community in making the coral more resilient can help us plan and incorporate these things into coral restoration, right? Like, so if we're doing coral restoration, we can test which we can collect these corals, bring them to the tanks, and then test which ones are more resilient. But you can also look at the symbiont community. What's the role of the symbiont community in there? And then if you see that it has a role, can we maybe infect corals from the same species to have a similar symbiont composition and then outplant them? So I think understanding, there's a lot of things that we need to understand still, like, but we see that the symbiont has a role. We saw like when exposed the corals to the heat stress, also other projects that I did for my PhD that I didn't show here for the purpose of time, but in any of this project, like you see an increase in the heat resilient symbionts when corals are exposed to heat stress. So like maybe what can we do with that information? Like, can we infect these corals with this heat stress? We know not all corals have the same symbiont composition. So understanding that maybe some species, that's not a strategy that's going to work, but just understanding the role of the symbiont community in providing resilience, I think that can be useful when we're planning for restoration events.
Excellent. We're getting lots of great questions here. This next question is a little bit different. It says, well, it starts with amazing talk. What is known about the mechanisms of environmental memory? How is it formed? Can it be lost over time? For example, will a transplanted coral become less resilient over time?
Good question. I don't think we have many answers for that yet, but we do see a memory, and it's not just on symbiont. Like we have, there are multiple mechanisms of memory. It's not just imprinted in the symbiont community. We also see that in the genetic of the coral, like in there are other mechanisms that we see the memory imprinted. I cannot see who wrote the question, but like I can follow up on that. I have like papers, we can discuss more specific details on that, but there are a lot of things that we don't understand. But I think that's a very interesting thing that I found in my study, that even after 2.5 years, like you still could see like a signature of the location where the coral came from. So like it means where they came from really has something imprinted in the gene of the coral, right? Like that made them respond in a similar way when they are exposed to some stress. So I think that's very interesting, and I don't think anyone has all the answers yet. Like there's a lot of things that we don't understand still about how this memory happens and how long it stays, right? Like it was from 2.5 years. You would imagine that for being such a long-term experiment, maybe it would be lost, but it was not. So how long can it stay until it's lost? I don't know. Can it be lost? I don't think anyone has the answers, but I think those are very interesting things to even consider for next like restoration or any project with corals, right? Like, is it worth to try to infect corals with different symbionts, as I said? Well, maybe it depends where they came from, right? Like if they have a memory, maybe they're not going to acquire a symbiont from that they're not used to. Like, so I think like there are a lot of things that we should take into consideration still and study still that we don't know about.
Excellent. This next question asks, what method did you use to specifically extract symbionts?
Yeah, great question. So the symbionts are inside the cell, so like they are microscopic. So you cannot identify them by naked eye. You have to do everything is through molecular work. So I did amplicon sequencing. So as I kind of described in one of the slides, I had a few slides about the multiple steps. So we extract the DNA and we do what we call library preparation. And then we use next generation sequencing to sequence. So we have millions of copies of the DNA of the symbiont in the coral. And then we use SymPortal. That's a platform that has been developed only to identify symbionts. So we can scientists that are doing similar things, we can standardize the pipeline, like all the bioinformatics is standardized by using Portal. And then after that, so I can see like which symbionts we have in each one of the corals and the number of each symbiont in the corals. But yeah, I use ITS to amplicon sequencing.
Excellent. Here's another question. Did these corals experience mortality during the 2.5-year experimentation period?
Yes, we had a lot of mortality, yes. That's expected. Like, I think it's just being in a tank is also stressful, right? Like, so when you brought these corals to the tanks, they had to be acclimatized. They were kept in the tanks for about like, I think like 1 month, a month and a half, like without any stress before we actually started the treatments. And we saw mortality, of course, like when we increase any stress condition, we saw a lot of mortality, and we saw mortality also different by species. So Postulopora, for example, we had very high mortality, like to the point that I didn't even show some of the results because of that species, because I mean, I had like very few representations like in the end of the experiment. So yes, we had mortality for sure, and the mortality really differed by species. Montipora captata, most of the samples survived. Postulopora compressa, most of the samples died. So we also see a difference in resilience according to species.
Excellent. We still have about 4 more questions. This next one asks, how do you see the natural selection will function over all this changes that ocean are passing?
How do you see natural selection? I think like, well, we can think in the ones that are more resilient are going to survive. I think you can think in the sense like the ones that have already higher resilience are going to survive, and the ones that are more affected, more fragile, like they're going to go away. And we actually see that already, like for some species, for example. So in Hawaii, we had the bleaching event in 2019, and we saw Montipora captata mostly bleached, but survived. We didn't see high mortality. Postulopora compressa, as I said, Postulopora, Postulopora acuta, we saw very high mortality in the end. Like, so they had a very high bleaching and a lot of mortality in the end. So like we expect the most, like for example, in Kaneohe Bay, a lot of Postulopora was decimated after the bleaching event. And Montipora survived. We had very low mortality of Montipora. So I think we can talk in these terms, like, so some species are gonna probably go away, some species are gonna resist. So when we say that corals are gonna die by the end of the century, we're really talking about different species, right? Like we're not gonna see a decimation of all the species. Some species are already more resilient and they might survive. But we're going to see for sure like a change in the ecosystem. Like some species are going to be completely gone. Like most of the individuals of that species might die, but some other species might persist.
Excellent. This one is asked, says, non-expert here, how do you assess whether or not the difference in coral responses are statistically significant?
Good question. Well, different things. So like in my dissertation, I was quantifying more, the Symbiont community, and then I don't think I presented a result on bleaching. Yeah, I had some results, but I didn't present that. But talking more about like, things that we are doing in the lab and how we are quantifying like how corals are responding differently to stress. So that can be done by color. So yeah, just like photographing corals before and after, and using statistical softwares to actually compare the different colors. That can be used by looking at the photosynthesis activity of the algae. So like in how the coral and the respiration of the coral, like, so we can with that get estimates of how stressed the coral is or not. And I think the Symbiont community also, like how many symbionts a coral has. So when corals bleach, they lose most of the symbionts that they have. So when they're about to bleach, they that is the symbiont. When the heat stress, when there is a heat stress event, the symbionts produce this toxin, this reactive oxygen that's toxic for the corals. So the corals get rid of the algae. So corals are going to have less symbionts when they are bleaching during a bleaching event. So we can also quantify, like in the microscope, like the number of algae that the corals have, and also looking at the type of algae. So like we know in a heat stress event, they're going to have more heat resilient symbionts. So also looking at the composition. So there are multiple ways that we can see how corals are responding.
Excellent. We actually have 3 more questions, but I'm going to actually ask only one of them. And for those of you who might have asked a question, these are all going to be forwarded to Mariana, who can answer them offline. I will point out to her which ones have not been answered. So for this last question, it starts with a compliment. Great presentation, Mariana. Although acidification seems not to have a direct effect on corals studied, would there be any possible indirect impacts? For example, could acidification change the other species, for example, fish, that interact with the reef system, and could that impact the corals?
No, for sure, for sure. I'm not saying that acidification is not going to have an impact in corals, and there are multiple people studying that. In my study, I didn't find that the algal symbiont was impacted by the acidification treatment. But we know there are studies like suggesting that acidification might be affecting the composition of the skeleton, might have an effect on the calcium carbonate, the skeleton of the corals. And I think even internally, I think, right, like for chemical reactions, like that might have an effect also. And for sure, it can have an effect on other species in the ecosystem that therefore would also affect corals. So I'm not saying that acidification might not be affecting corals, like the whole coral ecosystem, but in my study, I found that heat stress was the main driver of algal symbiont composition. I was not even looking at the health of the coral or like the physiology of the coral. I was actually looking at the symbiont composition itself only. So I found that the acidification treatment is not affecting the symbiont composition, but it might be affecting maybe the physiology, might be affecting other things that I was not looking at.
Excellent. So I'm going to go ahead and conclude this webinar. Audience, you've been fabulous. Thank you for the many really great questions. And Mariana, thank you for your presentation. I hope you continue to thrive in your fellowship and do this wonderful coral research. Caitlin, thank you for hosting and for your presentation in April, which I encourage the audience to watch. It's in the NOAA Central Library YouTube channel and waiting to be discovered and shared as well. Audience, I hope you'll join us again for a Knauss Lunch and Learn webinar, which we host every 3rd Thursday of the month at 12 p.m. Eastern Standard Time. So with that, I say goodbye, and I hope you all enjoy the day. Take care.
Thank you. Bye-bye.